Hello?
Can you hear me?
Yeah, I can hear you.
Oh, nice. Okay. I'm wearing my earpiece.
Oh, okay.
I've been having trouble with online things hearing me.
Right, right.
So,
Yeah, we've talked about that.
anyway, so just you and I again, or is anybody?
um,
I think Dick said it was at 10,
oh,
and it's 9.
I don't
So,
know. Yeah. Well, I mean, the time change, I don't know what happened, but yeah, I don't know. Anyways, it'll probably be, if you have something to present, that would be fine. There might be some other people coming, but I don't know yet. Um, why don't I, well, I can start off. Do you have something you want to present today?
no.
Okay, just checking.
I'm afraid I had a house disaster. I realized that the furnace needed to run because it's getting colder, and our basement flooded in the spring and stayed flooded for about a month and a half. And so basically everything that was on the floor had to leave and a
Uh-oh.
few other things. So,
Yeah.
yeah, so far it's 3 truckloads of things
Oh boy.
in
That's
various
horrible.
states of decomposition. Yeah.
That's horrible.
Yeah. No, it's not been fun. It's just a house disaster. Yeah,
Yeah.
I think the whole basement maybe just needs to leave in general.
Yeah. Okay. Well, sorry to hear
So
about that. Yeah.
I maybe I'd like to. There's been a couple of interesting things I've seen in passing, but um,
Why don't I get things started and then see who shows up, and we'll just kind of go from there. And
okay.
okay. So, welcome to the meeting. We have Susan's with us today. I might have some other people coming in, but a couple things I'd like to talk about today. So the first thing is, let me present my screen. Alright, so the first thing is that we are working on this periodicity in the embryo paper. And I know that Ojwal was asking about it, but he's unfortunately in quarantine right now for COVID-19. His father got COVID-19, unfortunately, and so he's been suffering with that. So best of
Well,
luck to him.
best wishes to his family and
Yeah,
hope everybody recovers.
yeah. So this is
I've had a scare too. Like my son had a headache, so he had to go get a test. They can't go anywhere until he gets his test results back.
yeah, yeah, yeah. Yeah, it's pretty rough still. So, um, so this paper is the periodicity in the embryo, emergence of order in space, diffusion of order in time, and we've talked about this in past meetings. So where we are right now on this is that we have an abstract. It can be revised. I've got it in Google Doc form. So last time you saw it, it was in the GitHub repository as a markdown file. Kind of getting it ready for heavier editing. So, and putting it into a Google Doc. And so, if you'd like to have permissions, I'll put this in the Slack channel, or I'll send it to people. I can even put it in the chat right now for Susan if you want to look at it.
Okay.
And so, yeah. And so it's about 8 pages right now. It needs to be probably about at least twice this length. And some of it will be like filling out some of the details about the analysis and that, but some of it is going to be like, the introduction, for example. So we have a nice introduction where we kind of talk through different parts of the paper, what are we talking about here? Why is it relevant? And then I have some graphs. So I have some graphs here, which are probably best in a formal results section, although I don't really have that distinguished yet. So a lot of this is just kind of like, I may need to put in like another, like we need to put in a method section, and then like define results. And then this is this is actually C. elegans. This is C. elegans. This one might actually go down into like down below this, but above synthetic embryo experiments. Okay, so this is Alright, so a little bit about that in a minute, but so we have results, and those involve C. elegans, of course, as we mentioned before, Zebrafish, which we have cell tracking data on, so we're doing these measurements of cell division and their frequency, and we're building these frequency plots here. That's what was that?
Sorry. For the salamander embryos, they're temperature dependent as well.
Okay.
As time, like they've definitely got a rhythm to their cell divisions, but they take longer to develop if it's cooler. Like it's time dependent, or I mean temperature dependent as well.
Right.
Just throwing that in there.
Yeah. Well, that actually might be something we can cite in the discussion, because we'll be talking a bit about why this, what, the relevance of this is. So maybe you could send some papers to me or the group about that.
Yeah. There's some really old papers like 1950s, 1940s, that sort of thing that I've looked at that, and I have a collection of them. It's sort of a hobby of mine, collect old papers about salamander eggs. So I can send some of that.
Oh, that'd be great. Yeah. Thanks. So yeah, that would be good. So we have the methods, the results. So this is a C. elegans, this is a zebrafish, different ways of plotting it out, and probably a little bit of discussion about what we're finding here. This one is an interval analysis of zebrafish embryogenesis. I think I have a corresponding one for C. elegans. I don't know if it's in here, but anyways. And then this is something we call information isometry, which is a method that we use that was developed in a paper several years ago by the group, where we look at taking like a lineage tree and flipping the nodes internally using different criterion. So you give the cells a different identity, and you flip the order of the tree and the subtrees within it, and then you based on the difference between like a control tree and like the tree that you flipped, what is the amount of information in each part of the tree. And so this is a lineage tree kind of like rooted here, and then the cell divisions proceed outward like this, and you can see the I think this has been done a little bit differently, so I'll have to like explain what's going on in this graph. Normally we use like a Hamming distance as an information measure. So if these cells, like if this cell is this cell here is in this position in the control tree, and it's flipped over here in the modified tree, then you have a Hamming distance, I think, of 2. And so and then you color code them in terms of their Hamming distance. And you can see like places where the tree has been displaced by that new criterion. We've used this with differentiation trees, but so we're ordering it based on like cell size differences between the daughter cells, but there are other criterion you can use as well. And so we might talk a little bit more about that. Then we go into what we call synthetic embryo experiments, which are these So the idea here is to take like a data, generate a synthetic data set of cell divisions in a lineage tree, and we're interested not in the position, between different nodes in the tree at different levels, but of the timing of the division. So now the synthetic embryo has, it's basically like a tree where you count each division event in the tree and you look at the distribution or the intervals between those. So it's a lot like what we're doing in this graph here for zebrafish, but we're doing it down here for like a synthetic example. And so we can start with a uniform set of divisions, maybe like every 20 minutes there's a division event. And then going from there, we can like distort the distribution of timing. So instead of every 20 minutes, we might say, well, the divisions occur at an unspecified rate, but it generally happens, within a certain interval of that 20 minutes. And so one of the things that you can do is you can generate these trees, or generate these data sets using a sort of a uniform distribution, but you can also generate them using a Poisson distribution. And the Poisson distribution gives you a little bit of variability in terms of the timing. So instead of every 20 minutes, it'll be like maybe 6 minutes, 8 minutes, 15 minutes, and so forth. And you can generate a distribution like that, and you can look at the difference between those two division regimes and see what they look like. And so that's what we've done here. I've given a little example in this table. So a small sample of our numeric simulation. All this needs to be like probably discussed in more depth. This is a frequency plot for the Poisson interval. So you have, this is frequency units of developmental time. So this is the number of, if you generate a bunch of these are the length of interval between division events in a Poisson distributed tree. And so you see that there's a sort of a cluster between 2 and 6. So most of the divisions happen between 2 and 6 units of developmental time, and very few happen after 10. So, we can look at it that way. And then this is kind of seems like it's not a very useful thing at this point. Other than knowing that you can do this and play around with the timing. But what does that actually mean, perhaps, biologically? And so one thing we can do is compare this with a uniform tree and look at the number of division events that occur over developmental time. So this Poisson distribution generates events, with a little bit broader variability than the standard uniform set of divisions. And so what we find is that after about 4 division events, you start to get more division events earlier in developmental time on average, so that you have this Poisson division time distribution that we've created here. Everything happens a little bit earlier than in the uniform case. And so that maybe means that there's some other type of distribution that it's using even to generate events earlier. You can make, you can have division events, I guess, like every minute, I guess. That wouldn't be that surprising, except that, there is this sort of, there are limits of like mitosis and things like that. So we just want to see what happens when you have this variability versus this uniformity. And so maybe if there, I don't know, maybe we need to put another comparison in here where it's like, it'd be like some other type of distribution.
I'd like to see that on that graph too.
Yeah. But we know that Poisson events occur quite commonly in nature. We know that, there's a lot of things that occur according to a Poisson distribution. So that's actually a pretty well established thing that might happen. But yeah, we can put other distributions on here.
I like to see the differences there.
Yeah.
That you're going.
So then this we kind of wrap the analysis up there. Then there's the discussion. So the discussion is really, this is where the more information about different species becomes like salient. So we recap what we've done. We've looked at 3 model systems: zebrafish, nematode, and a simulated embryo. We're looking at the tempo, what they call the tempo and the mode of development, which in evolution we know is something very famous. Stephen Jay Gould, I think, proposed tempo and mode as like a controlling feature of evolution, and this is associated with the famous punctuated equilibrium idea. And so, but then of course we're kind of asking a similar question for development. Are there tempos and modes of development that allow you to yield, to really explain what's going on in some of these data? So then we talk a little bit actually about something called the quantum mitosis hypothesis. And quantum mitosis is something that, I guess it's a theory that involves rounds of cell division and changes in gene expression, which then in turn have a potential to sort of modify the fate of different cells in the embryo. So quantum mitosis involves changes in gene expression. Every time there's a cell division, there's some change in gene expression. And every time there's a change in gene expression, that's potentially enough to change the identity of the cell and then ultimately the observed fate of the cell. So, you have a lot of changes in gene expression that change the identity of the cell, meaning like it's, not necessarily something you can see very clearly, like going from a stem cell to a neuron, but there are differences in like how, like say maybe a stem cell behaves on its way to becoming a neuron. So there's a lot of gradation there. We're not measuring that in this case, but we're interested in like those division events that might set up massive amounts of differentiation. And so, looking at these different waves might be a way to understand that, to see like, okay, there are a bunch of division events at a certain point in time. And then if you go back to the embryo, you find that, lo and behold, like sometime soon after that, you have a bunch of differentiation events where you get all sorts of tissues that are differentiated. And so that's we're going to add more to the discussion. We'll probably talk about temperature dependence and other things that, maybe things that we haven't accounted for here, or, things to look at and others, in the future. And then our references, we don't have a lot of references. We have about 15 references. So actually that can be gotten rid of, but yeah, so it's about 15 references now. So we need more references in this paper. But so that's where we are. I just wanted to give an update on that paper. It's pretty, it's pretty sort of scattered right now. It's not like, it doesn't look like a tight paper. It looks like it's got a lot of work to do, but that's okay.
It's a start, though. If you're at this stage, then you can get creative. That's always what I think. It's at that stage where you can really work with it.
Yeah.
I like this stage of the paper.
Yeah, it's due. I think we're shooting for the end of the year, so we've got about maybe 7 weeks to go on this.
Okay, I'll quickly find you at least the amphibian references.
Okay.
For you.
Yeah, thank you. Yeah, and so if you're watching this and you say, want to be involved still, then, we have other things you can do. Definitely literature search is one, and maybe, reading through and editing is another, like making sure things make sense. Suggestions for like graphs and visualizations is another. So we'll walk through all this, before it gets submitted. And hopefully, it looks pretty good by the time we're done, and looks like a good paper. But so that's a periodicity in the embryo paper. I think I have a thing on GitHub about like people's roles in the in the paper. I'm not sure what we had agreed upon for that. Oh, here it is. Periodicity. If I'm still sharing my screen, right? Okay. So in this repository, DevoWorm group meetings is periodicity. We have a to-do study plan, which is right here. And that is kind of spells out some of the things going on here. So people were expressing interest in the paper. And we have the data sets here. This will probably be something I'll need to put into the paper as well. But this will be for like the methods section. And so, yeah, we have this here as well. So if you want to consult that, you can.
Yeah, I've been, I don't have a whole lot of time. That's my problem.
Yeah.
Because I'm supposed to be doing mechanics and optical coherence tomography, and I have, like I said, a house disaster. And if I stray from any of that, I'm likely to get my supervisor annoyed with me.
Yeah.
We'll just say.
It's fine. It's,
Because
yeah.
Yeah, I am supervising the ball microscope already.
Yeah. There's Dick.
It's making some progress, the ball microscope. It's out for 3D printing now.
Oh, good.
Yeah.
Dick.
So yeah.
Oh, wow, that's your 3D ball thing. Okay.
Out for 3D printing. So we'll see if it works in a couple of weeks.
So I was just talking about the periodicity of the embryo paper that was that we're going to try to put into that special issue. So this is, I mean, it's not really that organized yet. It's sort of organized, trying to figure out how to put everything in place. But I'm just, kind of walking through it for people who are watching this. So it's like 15 references. We need more references. We need more interpretation and maybe a little bit of modification of the analysis. And so, I mean, there are a lot of things to do, but it's coming along.
Okay, good.
Yeah.
Okay. Yeah, I'm reading about the protozoa. The colonial ones sometimes have forms of synchrony
Okay.
in cell division. So it may be a very primitive feature.
Right.
I have a question from last week. Do Basilaria grow in Manitoba?
Yes.
Oh, they
But
do?
they're very hard to find. I had a student who spent a whole summer looking for them all over the place. And then finally she found them nearby at the end of the summer.
Oh, they grow at the end of the summer. Maybe that's the whole point. In August.
The guy in Germany, Thomas Harbich, he has periodic problems finding them or cultivating them. The cultures don't seem to last long. They slow down.
Oh, wow.
Yeah.
I found them in brackish water in Florida. So.
Yeah, I bet Florida grows lots of things.
Yeah.
So, yes, they're universal, what would you call them? Hide and go seek organisms. They always seem to be very hard to find when you want them.
Yeah.
Okay. So I was just wondering if I could put them in my 3D microscope if it works.
Well, the colonies are big enough sometimes. Part of the problem, in fact, is that if you want to do a big colony, you have to do a low power.
Oh, okay. Yeah, well, it's just, yeah.
That will give Bradley a hard time segmenting the cells.
Yeah. Yeah.
So,
So, yeah.
okay.
that sounds good.
Okay, looks like you got the wrong time for DevoWorm, huh? It's 9 o'clock.
It was, yeah, because of the time change.
But okay, yeah, okay. I put it down wrong. I will fix it for next time.
Okay, yeah, yeah. The time change is always weird because we get like that couple weeks where it's like trying to figure out when different time zones. It's hard to coordinate.
Yeah, I know.
Yeah,
that's why I gave up telling people what their local time was.
Yeah.
Let them figure it out from the UTC.
Yeah.
But I screwed up this time.
Yeah. So yeah, I guess next thing we'll do in the meeting here is I was kind of hoping Jesse would show up because we had a good discussion about note taking on Saturday at our other meeting.
Oh.
And he has a method he calls the twirly-gig method, and I don't know, like I guess or the twirly-bird method, like there are these things these seeds that spin around when they fall off the trees. And I'm not really sure what he's referring to. I mean, I guess it's like I guess it's something that he just kind of came up with, but I was going to go over some note-taking methods.
Okay.
So,
I need that. I need, because I have notes all over the place in every different type of format, and I lose them. Mm-hmm.
Oh. Yeah, I just start writing a paper. That's the only way I keep notes now.
Yeah.
Oh, really?
Yeah. Just start writing. Then I lose track of the papers.
I'll put them in EndNote. Yeah. Referenced.
Yeah. It's always a challenge because there are different methods that work well for people, and I don't want to speak to say that everyone has this, everyone has to do a certain method for note taking because it's and that's one of the problems that people have with note taking is that it's works for you well. It's like a diet, if it works well for you, then that works. If not, then forget it. And so,
Oh no.
Yeah, I mean, it's
this is kind of what I've been doing.
Oh,
multiple folders. I have 5 of these, and then there's 5 in one, and
I have about 50 of those.
Yeah.
So I have to go back to high school.
Yeah.
There's probably all sorts of good ideas in there, but I don't have time to read them.
Well, I have your laboratory notes.
Oh, okay. Well, your brain declines from high school down onward.
So, yeah, I guess that brings up a point that, there's a difference between taking notes in a paper and then digital notes where things are theoretically searchable, and so
You think I'm kidding? There are papers I wrote in my 20s that are now over my head.
Wow.
Oh no. That sounds like me and my math.
No.
Yeah. So.
Okay.
Yeah. If you assume. Is that?
You want to quit for today? Getting these Indian guys in looks like it's getting difficult.
Well, Ujjwal has. He's in quarantine for coronavirus.
Okay, with or without his computer.
Well, I don't know. They're having a rough time, and Jesse, I don't know where he is, but so I mean,
it's going up here. We had 400 cases over the weekend.
Wow. Yeah. People are going indoors for the fall. So.
Well, winter. There's snow on the ground here. It's light snowing right now.
Okay. Yeah. So I'll probably go back to note taking. So let's assume you have digital notes, but the issue, of course, is too if you prefer taking notes on paper, then there's no question of digitizing that. But if you want to take digital notes, there are a lot of ways you can do this. You can type things out in a document, they have now pens that will transcribe things into, like, from your writing to some sort of searchable text. But there are other types of note taking systems here. So the first of these is one of these systems is this system, better note taking. This is the point. What matters is better thinking. So this is the Evergreen note writing system, and this was created as a way to sort of like not only just kind of improve the way notes are taken, but sort of organizing them on the other end. You know, how do you sort through them after you've taken them? Because notes aren't really any good if you just like write them down and just put them away, and then you can't really access them later. So, you know, a vast majority of that writing fixates on a myopic life hacking type frame, which means that you're focused on answering questions like, how should I organize my notes? What kind of journal should I use? How can I make it easy to capture snippets of things I read, etc.? So that's, you know, that's a thing that people are thinking about, but they're not thinking about how to organize the ideas to maybe facilitate better thinking. So better questions are, what practices can help me reliably develop insights over time? How can I shepherd my attention effectively? So those are the types of things they're interested here. And then this led to this development of this Evergreen note writing system. And he calls it a fundamental unit of knowledge work, which means, I guess, Evergreen note writing helps insight accumulate. And so this is supposed to be like computer-supported thinking instead of computer-supported note-taking. So that's their focus on this. But so they've presented on this. This is a rather new method. This is a couple years old, maybe, where they're kind of building this note-taking system that includes context, feedback, and all these things that will be helpful in interpreting your notes as well. So, yeah, I mean, another thing, too, is like you might take notes and they might not be useful to other people. So if you take notes in some shorthand, you know, can you understand it later? Maybe in 2 or 3 years. Or 10 years. Yeah. Like we were talking about with the papers. But I mean, in this case, it's more like and I've done this, you know, write a note and then I don't know what it means later because I don't really remember the context or anything. So
reminds me of how I learned to comment computer programs.
Oh, yeah.
I wrote it a year later. I couldn't understand the program because I hadn't. And that's when I decided I better learn how to comment the program.
Yeah.
So
that's actually, yeah, that actually brings up the, so people talk a lot about like reproducibility in programming and in science, like methods. And this is one of the things they talk about is to have a method for sort of ordering sort of making sure that anyone can understand what you're doing if they revisit the work or if they see it for the first time. And so this is interesting in light of note-taking because we don't really think of note-taking as being reproducible. But that's actually what we're talking about here is reproducibility. And so, you know, if I can take a note and like pass it off to someone else, have a conversation with them or give it to myself like 5 years later and know what's going on, that's good. You know, I mean, it makes it worthwhile anyways.
Okay, look, I have a suggestion for that.
Yeah.
Mathematica allows pretty much free text inside a program. And since many of the things we do are computer programs, it allows much more than simple commenting. That you can actually have full text inside a mathematical program.
Yeah.
Okay, but I haven't used it. So.
I've used I've used it in MATLAB, and
Mathematica.
Oh, in Mathematica. Okay, well, I've used it in MATLAB, and yeah, you need to say, okay, I'm starting a new section, and this section does such and such, and you just comment it. I guess it's the same as computer programming.
Can you put a freeform equation in it, for example?
As long as it's between the comment bars or the stars, you're good.
Oh, you are? Okay.
Yeah.
Okay, so maybe that has that feature too. I'm just saying, since much of what your group does, Bradley, is programming, maybe the note-taking should be part of program development.
Yeah, I think that would be a good idea.
Okay.
Yeah. It's writing note here.
But that requires using a language that makes that easy.
Right.
Yeah, I had to take Mathematica off my computer because I have no space. I guess I needed a new computer. Just what I always wanted to do.
So a couple more. Yeah, so like if you go to GitHub, for example, you know, we take notes in GitHub and we use Markdown to sort of render things so that we can make, you know, notes editable. So if you want to work in GitHub, you can do this, and it's version controlled. But again, you don't have, and you can also do, you know, write a program and comment on it. So I don't have a example of that to pull up right now. But basically, if this window were filled with computer code and we had comments in it, you know, we could do some sort of commenting in maybe another file in the directory. So like, you know, instead of this, we'd have another file that said commenting or something else. So I mean, that's another way to go too. And you know, it makes it a little bit more, like, at least we have the version control aspect of it. So, you know, you can look at the history of a file, and you can look at something that maybe was talked about earlier and see how the comments changed, or people have addressed the comments. We were doing that a little bit with some of the writing that we were doing in another group of mine earlier this year. We were writing an essay, and we wrote, and we would have like comments embedded in the essay, and then, you know, we could address the comments and have versions of it where the comment, there was like a conversation in a comment. So we'd have like we'd do something like this. We'd have like 4 bars and then like a comment. And then if you commit it, you can see that it makes a comment. Like, this one doesn't work, but it's I can't remember if it's 4 bars or 5 bars. Anyways, there's a way to make a comment in Markdown where it sort of indents it and turns color, and then you can put, you know, notes in there, and they're kind of separate from the rest of the document. So those are all things that, you know, we can do in GitHub for note taking and annotation.
Do any of these languages, you know, many of these programs are hierarchically structured. Okay, are there any that allow you to open and close to a given level?
I know.
In other words, if I don't want to see everything between a begin and an end, can I close that?
Yeah, that's something you'd have to do, like in like HTML or CSS or something like that, where they actually do have ways of doing this, where you can build like a HTML document and then have like, you could open and close different parts of it. But it requires, it's not something you can do like in a very casual note-taking environment, at least not that I know of.
Yeah, there used to be a word processor that made that extremely easy. It was called MindWrite. And I actually made a mixture of MindWrite and Pascal so that I could take the hierarchical structure of a Pascal program and easily incorporate it into a MindWrite document. And go back and forth between the two.
Yeah.
Okay. You might look up MindWrite. It might be a language worth resurrecting. It's much easier to do than the hierarchical stuff in Word.
Yeah, that's true. You can do that in Word, but it's
clumsy in Word.
Right. And
that was MindWrite was around 1980.
Okay. Do you spell mind right, like R-I-T-E or I-G-H-T? Okay.
W-R-I-T.
Or W-R-I-T-E.
The university was looking for feedback as to how they could support people doing visualizations. And I just said, well, MATLAB comes to mind. Why don't you have it available basically for students instead of having to buy it? Anyway.
Yeah. So that was, I think we've had a nice discussion on this with this, and then So then this is something called a Zettelkasten. So I tried to build a Zettelkasten following Andy Matuchik. This is the guy who developed this Evergreen system. And now I have this weird thing. So this is where they talk about this book, How to Take Smart Notes by Sanke Ahrens. And this is a book about, where they give you a very broad framework for taking notes, and then you have to implement it yourself. So this is a method called Zettelkasten. As I built one, I kept saying to myself, this would be a lot easier if I saw what good Zettelkasten actually looked like. So I decided to design one in software. You saw Andy Matuchik's notes, and this is, this Evergreen is an example of the Zettelkasten, but doing this digitally. And so this article, and I can put a link to it here in the chat if you want to read more, but they kind of go through implementing this on their own. So the idea here is that, people talk about this as a framework, but can people use it in a way that's makes sense to them? And so this is kind of exploring this. So how Andy Matuchik creates Evergreen notes. So there's a process here: reading inbox, transient notes, writing inbox, Evergreen notes, speculative outlines, and writing. And then the Evergreen notes are, the principles behind them are that they're atomic, concept-based, connected, synthesized, and distilled, and then have titles and things like that. So he kind of walks through these points. So I mean, I don't know if I'll go into that too much more, but if people want to read that, I'll put up a link. I'll send a link out on Slack as well for that. Then there's this taxonomy of notes, which is this is a blog post. So this talks about taking smart notes. It's one of the most efficient ways to increase your productivity and creativity. Well, what kind of notes? And so this is they're attempting to build a taxonomy of notes here, which include ephemeral scratchings, prompts, and incomplete notes, as well as evergreen notes. And then someone here shared 4 levels of evergreen notes. So they actually have figured out different types of evergreen notes based on Ames type sources of notes. And so this goes back quite a ways. This goes back to 1962 in a book about study methods. They proposed this sort of taxonomy of notes, separating note-taking from note-making. And so, let's see. Oh, this is a nice diagram here where it kind of shows sort of the inputs and outputs of notes. So you have all these different inputs, like the structure and the temporality, which is like the time input, the goals, the methods, and then outputs are format, medium, and source.
Okay, Bradley, there's one other thing you might want to look at. There are programs that are sold for organizing laboratory notes. Okay?
Right.
It used to be that people settled priorities by laboratory notes, and I think this takes that into account.
Yeah.
Okay? Remember, I don't know if you know that stuff, the old sealed letters to a president of a society. open this in case you think I'm scooped.
Oh.
Those are from the 1700s and 1800s, I think.
Oh, okay.
But now there are digital versions of this kind of stuff.
Oh, okay. I'll have to look into that.
There are probably a few products.
Oh, yeah, I'm sure people have maybe revisited this or maybe not. That'll be interesting to see. So then I put a link to the taxonomy notes in here. And then, yeah, thank you for the conversation on this. I think maybe we'll follow up on this next week or so. Maybe Jesse, if he's here, will have some things to say about it. But I think it's worth talking about because I think it's a common challenge to a lot of people, and it's quite useful.
I could send you a couple of boxes of notes to scan.
Oh, no. That would be, it wouldn't be as useful, but
those notes where you don't understand them anymore would be intriguing. There would be a puzzle, lovely puzzle.
Maybe we should go into the category of write-only notes.
yeah.
Yeah, you know what read-only means.
Right.
You can only read it. You can't change it. Write-only
Notes.
you can't.
I have some of those that I take them in a hurry while someone's talking. Yeah.
There you go. Okay, so you're gonna. There, we got you going now. You can write a satirical review of note-taking.
Yeah. So
Most of mine are write-only.
Yeah.
That's why I start with papers, because at least the paper I might eventually get around to publishing it.
Yeah. So last thing I wanted to talk about today was a couple papers that we've run across, and I'll kind of go over these quickly, probably before the end. Try to get it within an hour here. So we've got a couple of interesting ones. First of all, there's a video here of something called Neuro Kernel, which is where they're emulating a Drosophila brain on multiple GPUs. This might be interesting to some of the people working on or interested in connectomics and some of these topics. So this
can I make a comment here?
Yeah.
If you want to go to primitive neural systems, this old book I'm reading continually talks about neural networks inside single cells.
Oh, really?
Yeah, so various protozoa. And of course, these are not nerves because it's inside one cell.
Right.
And this is a 1940 book, so it's not germane to what I'm reading it for, but I keep noting this continual talk about them having these neural networks.
Wow. Yeah. What is do you know what the name of the book is, or?
Yeah, let me get it just a second.
Yeah. Yeah, that'll be interesting. So, this video is the INCF put it out. It was a talk. And then this is about them simulating a Drosophila brain, I think, cell by cell and by connections. And so this video goes through a lot of the stuff. They go through, it is an open source project, so they have, very open structure to it.
There you go.
The invertebrates. Yeah.
Invertebrate. It's volume 1. I have 6 volumes. Whoops, who's it?
The invertebrate.
Come on.
Yeah, the
By
invertebrates.
Hyman?
The Invertebrates by Hyman. Yeah, 1940.
Okay. Well, that's good, yeah.
And I have no idea. I'm reading this old stuff, and then I'll use the names to go forward with the literature. And the names of the organisms.
Right.
But I'm doing it from the point of view of the boring billion.
Oh, okay. Yeah.
Okay, I just noticed there's a lot of stuff about as if these guys have. Well, how should I say? It looks like, you know, the old-fashioned way of signaling around a house by having a rope on pulleys that went from one room to the next?
Yeah.
Okay, it looks something like that.
Oh, okay.
Now, you have to realize 1940 is before the discovery of microtubules, before the discovery of DNA, before the discovery of genomic-based phylogeny.
Yeah.
Okay, but it's rich in observations of behavior of these organisms.
Yeah. Yeah.
So in that sense, it's worth reading, but then I have to follow it up.
Right.
See what else was done later.
Yeah. So, yeah, thanks for that. So this video, it's available on YouTube. They talk about, you know, the entire sort of Drosophila brain. So we know that the C. elegans brain, we have the full connectome, and they're rapidly finding or releasing full connectomes in different parts of the Drosophila brain. So I think they have a published connectome of one part of the Drosophila brain. I don't think it's the mushroom bodies, but it's something else. And so they're able to sort of give us some of these. They're using that data as the basis for their simulation here, and they're able to do. I mean, I'm not gonna go through the video that much, but if you want to see it, there it is. A couple other papers here. So this one is the morphostatic limit for a model of skeletal pattern formation in the vertebrate limb. This is an interesting mathematical modeling paper. It's from 2007. I ran across recently where they have this a recently proposed mathematical model of a core set of cellular and molecular interactions present in the developing vertebrate limb was shown to exhibit pattern forming instabilities in limb skeleton-like patterns under certain restrictive conditions, suggesting that it may authentically represent the underlying embryonic process. And so they do use a model. What was that?
Sorry, I have an addition for that.
Okay.
Dr. Jane Evans from the University of Manitoba observed that lung development caused limb development. If you didn't have lung development, you wouldn't have limb development. Something about they are linked.
Wow. Okay. Well, that's interesting.
Okay.
Yeah.
Here's the original connectome. I just gave it to you.
Let's see. This one here. Oh, yeah, the bones. Yeah.
Oh, you're old enough to know that song, huh?
Well, I've heard of it. Yeah, I mean, it's, yeah. So it's like the bones are connected. One bone is connected to the other. And yeah, that's, yeah.
The refrain is, now hear the word of the Lord.
So they have this, yeah, so this is then they develop a mathematical model for this. And so this might be an interesting one if you're interested in some of these issues related to limb development, which is an interesting part of development we don't talk about as much in the group as we usually do, because it's not really a C. elegans thing, but,
okay.
You know.
Let me ask you a stupid question.
Yeah.
Connectome between cells?
Well, there's what do you mean, like connectome between?
You identify a specific cell and you say it's connected to another specific cell.
Yeah, so yeah, usually the way they do the connectome identity is sometimes they'll use like gap junctions, and if they can, you know, they'll get like micrographs and they'll look at the gap junctions between cells,
okay.
Or sometimes they'll use.
Gotcha. Has anyone looked at the change of the cellular connectome over time?
Just cells, you mean like if they're connected to one another somehow through.
Do they change connections to different cells?
Yeah, over time, yeah. I mean, that's one of the things in C. elegans, for example, in development, we kind of know that, like, if you look at it like neurons synapsing onto other neurons, we know that there are changes over time in some of those, in some cells, but not others. With respect to gap junctions, it's more conserved, so you don't see a lot of changes in development, but it's, we don't really know that. So it's an interesting topic, but it hasn't really been explored very much. So, but it has a lot of implications for even like cell-to-cell communication. If cells kind of move around the embryo or around the developing, phenotype.
Yeah. In fact, one of the distinctions between plants and animals is that people presume that because of the cellulose binding, plant cells don't change connectome. And I don't think that's correct.
I
think
was
it's
reading something
especially. Okay.
just that the connectome changed type on the fly in a cell. They changed from a gap junction to another type of junction.
Oh,
I don't think they captured that paper.
okay. No, my guess is that at least in plant meristems, where the growth is occurring, that there could be a lot of changes of cell position and cell contacts.
Yeah.
Okay. That makes sense to me.
Maybe it makes sense, but it's contrary to the dogma.
Like that. That's not where I read things. Not lately.
So I'll finish up with these other two. I'll just mention these other two papers quickly. This actually, going back to this first paper, they actually do some mathematical modeling in something they call Turing space. So we identify regions in the parameter space of a reduced system where Turing-type pattern formation is possible, which we refer to as Turing space. So they're actually able to get a good idea of with these simulations, what areas of that space are amenable to sort of Turing pattern formation. So then related to that, though, there's another paper on studying the evolution of the primary body axis in vivo and in vitro. And so this is related in the sense that there's this metazoan body plan is established during early embryogenesis by a collective cell rearrangements and evolutionarily conserved gene networks. And they're focusing on gastrulation here. And so while substantial progress has been achieved in terms of characterizing the embryonic development of several model organisms, the underlying principles remain enigmatic. And so despite the diversity of pre-gastrulating embryo and adult body shapes across the animal kingdom, the body axes, which are arguably the most fundamental features, generally remain identical between phyla. So they go on to talk about this. They review key examples and propose that similarities in morphogenesis, as well as associated gene expression dynamics, may reveal an evolutionarily conserved developmental mode, as well as provide further insights into the role of external or extra embryonic cues in shaping the early embryo. So embryo-like systems can be employed to inform previously uncharted aspects of animal body plan evolution and patterning rules. So we have a comment in the chat here, I think. Okay, so this is...
this is LOM, I think, had a commercial product for lab notes.
Oh, okay.
Okay.
All right. Thank you. All right. So check into that. So that's the paper, this second paper, and so this is another interesting paper, which we don't have a lot of time to go over. But they give a nice diagram of primary axis formation during gastrulation metazoan embryos and corresponding artificial systems. So they're looking across phylogeny here of metazoans from Porifera, cnidarians, and then into arthropods, and then mollusks, and then into vertebrates finally. And so you can see some of these changes as they exist in embryos across phylogeny. And this is sort of the idea of the paper. I don't think they don't introduce a mathematical model here, but they get into a lot of interesting topics, like, self-organization. They talk about that.
Yeah, you can see what's missing here.
What?
Protozoans.
Okay. Yeah.
But some protozoans undergo gastrulation-like movements of the surface of the cell.
Yeah.
Okay. Okay. What? Can you paste the title of that paper in, or what's the easiest way for us to get a copy of these?
Well, I can paste the link to this directory.
Oh, okay.
And I don't think I did that yet. So let me do that. I'll put that in here, and it should be shared. If it's not shareable, request it, and I'll do that. And then let me look at this final paper here real quick. And this is Disentangling Scaling Arguments. So this is an interesting paper. If you think about scaling, and this is more mathematical than developmental, but if you think about like scaling, like morphological scaling or some other scaling law that they often talk about, like in network science or something, then this is something that developed in the field of physics. They talk about like power law scaling and things like that. So this paper kind of goes over, contemplating what that means, and then also talking about maybe, where it is and isn't applicable. So it's a nice kind of it does go over a little bit of the history, and it goes over a little bit of the mathematics of it. And so, I mean, if, instead of when you read about power laws and things like that, sometimes it's good to have a good background for that. And this is the paper for that. So you can read a lot about, scaling laws in different physical systems and how, they're calculated and what they mean. And so I'm not going to get into this paper because it's just a comment, but that's also in there as well. So I think we're at the top of the hour. So I would like to thank everyone for joining us this week. And I think we had a nice discussion about the note-taking and about embryos and everything else. And this for the paper that I showed you. That's an ongoing work. Like I said, we're targeting submission at the end of the year for that. So if you're interested in participating, if you want to, we'll probably send out drafts when we get closer to that point. But that's something everyone can help with. But yeah, if you have ideas for it, or if you want to see the draft in its current state, I'll send you a link to the Google Doc, and maybe we can discuss it more next week too.
Okay.
All right.
Bradley, can you? I can't get into that Google Drive.
Oh.
Requires a password, which required a code that went to a cell phone, which I don't have.
Okay, I'll just send you the papers. I'll send the papers out by email. It should be fine.
Okay, thanks.
All right, thanks for everything. All right.
Okay, bye.
Okay, talk to you later. Bye.